Welcome back to my channel. In this video, I will compare two widely used cloning vectors, that is PBR322 and PUC19. First, let's check what cloning is and why we use this technique. In cloning, we need three components: a piece of foreign gene, a cloning vector, and a host cell. First, we will digest the cloning vector and the foreign DNA with the same restriction enzyme. For example, if we treat the foreign DNA with BAMH1, then we have to treat the cloning vector with BAMH1. Now you can see in both cases the same sticky ends are generated. If we mix and incubate these two fragments, they can join together in the presence of another enzyme called DNA ligase. Now we will use the artificial transformation technique so that the E. coli cell will take up the DNA. The cells are then plated and incubated. When the cells grow, they will also replicate the plasmid DNA, so in the end, we will get multiple copies of the foreign DNA. I hope now you can understand the role of cloning vectors in DNA cloning. A cloning vector is a small piece of DNA. It can be linear or circular, or it can be single-stranded or double-stranded, that helps to replicate and stably maintain the foreign DNA inside suitable host cells. Both PBR322 and PUC19 are plasmid cloning vectors that were designed in the lab for cloning in E. coli. The PBR322 was reconstructed by Bolivar and Rodriguez in 1977. It was originally constructed by ligating together restriction fragments from three naturally occurring E. coli plasmids, that is R1, R6.5, and PMBL1. PUC is the updated version of PBR322 and was generated in 1985 by the researchers from the University of California. To be an efficient cloning vector, it should be small. The size of PBR322 is 4.3 kb and PUC19 is 2.6 kb. The transformation efficiency will drop drastically if the total size of the plasmid vector is larger than 15 kb. So PBR322 can accept 10.7 kb and PUC can accept 12.4 kb. Both plasmids contain an origin of replication that can be recognized by E. coli DNA polymerase. ROP will control the copy number of the plasmid, and more replication means more number of copies of the DNA we will get. For PBR322, the copy number is 15 to 20 copies per cell, and PUC19, it is 500 to 700 copies per cell. To insert foreign DNA, several restriction enzyme sites should be present. More number of restriction sites means more choices are there. For efficient cloning, there are over 40 enzymes with unique cleavage sites on the PBR322 genome and 32 unique restriction enzyme sites in PUC19. Out of 32, 13 unique sites are present in this small region called multiple cloning sites, or in short, MCS. Now, the last problem is, after the digestion and ligation reaction, how we can select those cells only which uptake recombinant plasmids. Because of the complementary nature of the sticky ends, plasmid can also self-ligate, and not all cells will take the foreign DNA. This problem can be overcome by inserting some selectable markers like ampicillin resistance and tetracycline resistant genes in PBR322. Out of 40, 13 restriction enzyme sites are present within tetracycline resistant genes and 6 sites are in ampicillin resistant genes. If we digest the PBR322 with BAMH1, it will cut here. During ligation, two possibilities are there. It will self-ligate or it contains the foreign DNA here. When it is self-ligated, the reading frame of the tetracycline gene will be restored, but here it is lost and inactivated. This phenomenon is called insertional inactivation. Now let's check how we will discriminate self-ligation versus insertional inactivation. We will add ampicillin to one plate and add tetracycline to another plate. The addition of antibiotic into the medium will kill all the bacteria that do not uptake any plasmid. Now if the bacteria are ampicillin resistant, it means they uptake the plasmid. We will use replica plating to check all those bacteria are tetracycline resistant or not. If the bacteria are tetracycline resistant, it means the plasmid is self-ligated. If they are tetracycline sensitive, that means it contains my gene of interest. The same thing is applicable for the ampicillin gene also. Now, we will check PUC19. Here the ampicillin gene is present as a selectable marker. In addition, one reporter gene called beta-galactosidase is also there. We will see the difference between a selectable marker and reporter gene later. Multiple cloning site is present in the reading frame of LAGZ. Now I will cut the vector in this area and try to ligate my gene of interest here. It will inactivate the beta-galactosidase enzyme if not self-ligated. Now we have to prepare the medium and we will add ampicillin, IPTG and XGAL into the medium. If the bacteria do not uptake any plasmid DNA, they will die because of the antibiotic. If the plasmid is self-ligated, the LAGZ gene is intact. And IPTG will induce the LAGZ gene to produce more amount of beta-galactosidase that will degrade the chromogenic substance XGAL and produce a blue color colony. So blue color colony means the bacteria are not recombinant. But if the plasmid containing my gene of interest, it will alter the reading frame of the LAGZ, resulting in LAC-negative cells. The recombinant cells can't break down the XGAL and the colony will be white. This process of recombinant selection is called blue-white screening. The researcher can visually distinguish the recombinant versus non-recombinant cells very easily. I hope you like this video, so don't forget to press the like button, and if you are watching my video for the first time, then press the subscribe button and the bell icon. And lastly, thank you all.